Okay, let's unpack this. When we talk about the gastrointestinal tract, most of us probably think of, well, just digestion, right?
Yeah, that's the common view.
But what if I told you this incredible system is actually your body's largest immune organ, a master of complex communication, and even houses its very own second brain?
It's truly fascinating when you dig deeper.
Today, we're diving deep into the functional anatomy and the intricate principles of regulation within the GI tract. Our goal for you, our dedicated listener, is to not just scratch the surface, but to really master this complex subject.
Yes, understand it with the depth of an elite professional, really.
To truly grasp how our body processes food and nutrients, and frankly, so much more, we absolutely need to go far beyond the basic textbook overview.
Exactly. This deep dive is crafted to explore every layer, every connection, every intricate control mechanism in detail. You'll finish this conversation with a comprehensive understanding that truly sets you apart. You'll really have a handle on it.
We're drawing all our insights from a really comprehensive source on human physiology. Think of this as your shortcut to mastery. We're extracting the most important nuggets of knowledge and surprising facts to make you incredibly well-informed on this fascinating system.
Let's get into it.
So let's start with the big picture. What is the GI tract and what are its core functions?
Right, the basics first.
At its heart, the gastrointestinal tract is the alimentary tube running all the way from your mouth down to the anus, plus all the crucial glandular organs that contribute their contents along the way.
The whole passway.
Its main job, at first glance, seems straightforward. Absorb nutrients and water into your bloodstream and get rid of waste.
Seems simple, but it's not.
To do this, it relies on four major physiological processes working in concert: motility, secretion, digestion, and absorption.
Those four pillars, yeah.
Digestion itself involves both physical breakdown, chewing and churning, and chemical modification of food macromolecules so they can actually be absorbed.
Breaking it down small enough.
And the GI tract, along with its associated organs, secretes a host of things: enzymes, biological detergents, ions, all creating the perfect internal environment for these processes to happen efficiently.
Setting the stage chemically.
And here's the kicker. All of this is incredibly tightly regulated to maximize efficiency.
Regulation is key. And what's really crucial to understand is that the GI tract is far more than just a digestive tube.
Right, you mentioned the immune function earlier.
Exactly. It's truly a critical line of defense, representing the largest immune organ in your entire body. Huge. But its functions extend even further. Consider its role in drug absorption.
Oh yeah, like taking pills.
Right. Many medications, whether taken orally or rectally, rely on the GI tract to enter your system. It's also a major player in excretion, not just for undigested food waste, but also for products from the liver.
Things the body needs to get rid of.
Precisely. Things like cholesterol, steroids, and lipid-soluble drug metabolites. So it's constantly managing both what comes in and what needs to go out, and it's doing a remarkable job of protecting your internal environment from the external world.
That's a huge perspective shift, realizing it's also a frontline defender.
Wow.
Okay, now that we've got that grand overview, let's really zoom in on its physical blueprint, the functional anatomy itself.
Let's look at the structure.
The alimentary tract is a long, continuous tube with distinct segments, each with specialized roles. Starting at the very beginning, we have the mouth, then the esophagus, stomach,
The upper parts.
and what we call the small intestine, which itself is divided into three parts: the duodenum, the jejunum, and the ileum.
Duodenum, jejunum, ileum. Got it.
Following that, we get to the large intestine, comprising the colon, rectum, and anus.
Finishing the tube.
And it's not just the tube. We also have crucial associated glandular organs, like your salivary glands and the exocrine pancreas, both secreting vital digestive fluids into the gut lumen.
Adding the juices, essentially.
You even have these fascinating little tucked-away structures called invaginations, blind-ending glandular structures like Brunner's glands in the duodenum.
Oh, Brunner's glands, right.
They secrete copious amounts of bicarbonate.
Yeah, HCO3, which is a key part of neutralizing stomach acid as it enters the small intestine. Really important buffer.
Makes sense. You need to protect that delicate small intestine lining.
Precisely. What's particularly insightful here is the incredible regional specialization along this tube. Each section is optimized for its specific tasks.
Okay, like how?
Take the stomach and the colon, for instance. They function primarily as key storage organs for processed food, what we call chyme or waste.
Holding tanks, sort of.
Exactly.
Yeah.
Their shape, size, and even the type of smooth muscle they possess, which produces sustained tonic contractions, are all designed for efficient containment. The small intestine, on the other hand,
That's where the action happens.
that's where most digestion and absorption happens, yeah. Its major specialization is an incredibly vast surface area, which is absolutely vital for efficiently absorbing nutrients.
We'll probably get into how it achieves that later.
Oh, absolutely. Villi and microvilli. Then the colon takes on the crucial role of reabsorbing water and ions, preventing dehydration.
So, reclaiming water before waste is eliminated.
Right, and all this ingested material is precisely moved along by the coordinated action of muscles in the walls. And separating these distinct regions are specialized muscle structures called sphincters.
Like little gates.
Muscular valves, yeah. They act like muscular valves, isolating regions, selectively retaining contents where needed, and preventing any backflow. This ensures the one-way, efficient flow of digestion.
Keeps things moving in the right direction.
Keeps everything orderly. Now, beyond the tube and its muscles, the supply lines are absolutely vital for the gut's function. The blood supply.
Gotta nourish the system itself.
Right, but it's unique. Unlike most other organ systems, the venous drainage from the GI tract doesn't return directly to the heart.
Oh, where does it go?
Instead, it first goes into the portal circulation, leading directly to the liver.
Ah, the liver gets first dibs on everything absorbed.
Pretty much. This means the liver receives a substantial portion of its blood supply directly from the gut, rather than just arterial flow. This arrangement is crucial for processing absorbed nutrients and, importantly, detoxifying any harmful substances before they reach the general circulation.
That makes so much sense. A built-in checkpoint.
It really is. Then there's the lymphatic drainage. This system is incredibly important for transporting large, lipid-soluble substances.
Fats and things.
Exactly. Things like dietary lipids, certain fat-soluble vitamins, and some large drug molecules. These are simply too big to pass into the tiny blood capillaries.
So they need a different route.
Yes. They enter the lymph vessels within the intestinal wall, eventually draining into larger lymph ducts and ultimately into the systemic circulation, bypassing that initial liver pass for the most part.
Interesting. So fats take a different path initially.
Mm-hmm. This has major physiological implications for how your body handles fats and how certain orally administered drugs are delivered and metabolized.
That distinction with the portal circulation and the lymphatics is a massive piece of the puzzle, explaining so much about nutrient and drug processing. Okay, if we could metaphorically use a scalpel and zoom in even further.
Microscopic.
Yeah, let's explore the intricate cellular architecture of the gut wall itself. When you look at the tubular gut, you'll discover it's organized into 4 primary layers.
Or main layers, right.
From the inside, closest to where the food passes, we have the mucosa, then the submucosa, followed by the muscularis externa, that's the muscle layers, and finally the serosa, which is the outermost protective layer.
Mucosa, submucosa, muscularis externa, and serosa. Let's really peel back these layers to understand their unique contributions.
Okay, start from the inside out.
Makes sense. The mucosa is the innermost lining, directly interacting with the contents of the gut. It's actually composed of 3 sublayers: the epithelium, the lamina propria, and the muscularis mucosae.
3 parts to the mucosa itself.
Okay. The epithelium is a single layer of specialized cells that forms the very surface lining the lumen and also makes up the glands embedded within the mucosa.
The actual contact surface.
Exactly. And within this epithelium, you find a diverse array of cell types, each with a specific job. The most abundant are the absorptive enterocytes.
Entero meaning intestine, cyte meaning cell.
Precisely. They're essentially the food processors, packed with proteins designed for digesting and absorbing macronutrients. Then there are the enteroendocrine cells, EECs.
Hormone cells.
Right, they're like the gut's hormone dispatchers, secreting regulatory peptides and amines to send messages throughout the body. We'll talk way more about them.
Hey, that.
In the stomach's lining, specifically, you have cells specialized for producing protons or stomach acid. And throughout the GI tract, mucin-producing cells are always at work.
Making mucus?
Creating a protective glycoprotein called mucin, yes. It forms a crucial lubricating and protective barrier against, well, everything passing through.
Very important layer.
Absolutely.
Yes.
And what's truly remarkable is how the epithelium's specialization varies by region. For instance, the intestinal epithelium is explicitly designed for absorption, with highly selective uptake of nutrients, ions, and water.
Max efficiency.
It achieves this incredible efficiency through finger-like projections called villi, which dramatically increase its surface area.
That's the tennis court analogy, right?
That's the one. It's a massive surface area increase. In contrast, the esophageal squamous epithelium is more like a simple conduit, providing protection from rough food without any significant absorptive role.
Different job, different structure.
Exactly. These villi, along with crypts, which are invaginations or folds in the epithelium, collectively contribute to that massive surface area essential for absorption.
So villi stick out, crypts go in.
A good way to picture it. And here's something mind-blowing. The entire GI tract epithelium is continuously renewed by rapidly dividing stem cells located in the crypts.
Constantly rebuilding itself.
Constantly. This entire lining gets replaced approximately every 3 to 5 days in humans. It highlights its incredible regenerative capacity, but also its vulnerability if that renewal is disrupted.
Wow. 3 to 5 days. Incredible. What's under the epithelium?
Beneath the epithelium is the lamina propria. It's a loose connective tissue layer that's rich in glands, tiny lymph vessels, those lacteals we mentioned for fat absorption capillaries, and nerve fibers. It's like the support structure right beneath the surface.
Immediate backup.
Yeah. And the thin muscularis mucosae is the innermost layer of smooth muscle within the mucosa itself. Its contractions cause the folds and ridges we see in the mucosa, further enhancing its functional capacity, especially for absorption. It kind of stirs things locally.
Okay, so that's the 3 parts of the mucosa. What's next?
Moving outward, the next layer is the submucosa. This layer also consists mainly of loose connective tissue, with a meshwork of collagen and elastin fibers providing elasticity. In some regions, like the esophagus and duodenum, you'll even find glands located within this layer itself.
Providing structure and some glands.
And housing another important nerve plexus we'll get to. Next, we have the muscle layers, often called the muscularis externa or muscularis propria.
The main muscles for movement.
Exactly. This typically consists of 2 substantial sheets of smooth muscle cells, an inner circular layer where the muscle fibers wrap around the tube.
Squeezing it.
Right. And an outer longitudinal layer with fibers running along the length of the tube.
Shortening it.
And the combination creates complex movements. What's truly fascinating is that nestled between these 2 muscle layers lies a critical nerve network called the myenteric plexus, also known as Auerbach's plexus.
Nerves embedded right in the muscle.
Controlling it directly. The powerful, coordinated contractions of these muscle layers are what enable the mixing and circulating of the contents within the lumen and, crucially, propel them along the entire GI tract peristalsis.
That propulsive movement.
Right. We should also briefly mention the submucosal plexus, or Meissner's plexus, located within the submucosa.
Another nerve network.
Yes, primarily controlling secretions and blood flow and interacting with the myenteric plexus. These intramural plexuses, myenteric and submucosal, collectively form the enteric nervous system.
The second brain we mentioned at the start.
That's it. It's incredibly complex and capable of independently integrating motor and secretory activities. A whole nervous system just for the gut.
Amazing. And the final layer.
Finally, the serosa, or adventitia in some parts like the esophagus, is the outermost layer. It's composed of smooth, squamous mesothelial cells and is actually part of the mesentery, the tissue that lines the abdominal wall and suspends the organs.
Holding everything in place.
And lubricating it. The serosa secretes a thin, viscous fluid that lubricates the abdominal organs, allowing them to move freely and glide smoothly past each other as the muscle layers contract and relax during digestion. Reduces friction.
That cellular architecture is truly a marvel, every layer with such specific roles. Now, let's shift our focus from the physical structure to the dynamic control systems.
How it all works.
Yeah, the master orchestrators that make all of this precise function possible. The GI tract is fascinating because it swings between periods of relative quiet, like between meals,
Basal state.
and then bursts into intense activity after food intake, postprandial state. This means it has to be incredibly intelligent about detecting and responding to food.
It needs sensors. Lots of them.
It also needs to accurately sense the macronutrient content of a meal, how much fat, protein, or carbs, and then mount the most appropriate physiological responses.
Tailoring the response to the meal.
All while communicating seamlessly with associated organs like the pancreas. And because it's such a long tube, there must be sophisticated mechanisms for communicating events from one end to the other, what we call proximal distal communication.
All right. What happens up top needs to influence what happens further down.
Exactly. How does it manage all that?
It's remarkable how the GI tract manages such dynamic and precise responses. It relies on three sophisticated principal control mechanisms working in concert: endocrine, paracrine, and neurocrine.
Hormones, local signals, and nerves.
Essentially, yes. Each of these systems plays a unique and vital role in ensuring optimal function.
Let's start with endocrine regulation. That's hormones traveling through the blood, right?
Correct. Messages travel far through the bloodstream. In this process, a specialized sensing cell in the GI tract, usually an enteroendocrine cell or EEC.
Those hormone dispatchers again?
Exactly. They detect a specific stimulus, say certain nutrients like fats or proteins in the lumen. In response, they secrete a regulatory peptide or hormone
Into the bloodstream.
into the bloodstream, yes. This hormone then travels via the blood to distant target cells that express very specific receptors for it.
Lock and key mechanism.
Precisely. These GI hormones can influence other regions of the GI tract, communicate with associated glandular structures like the pancreas, and even impact other tissues, including the liver, muscle, and significantly the brain, affecting things like appetite.
Wow, gut hormones talking to the brain.
A major area of research, the gut-brain axis. Now, there are open-type EECs, which have an apical membrane directly exposed to the gut lumen for sensing its contents, and a basolateral membrane for secretion into the blood or tissue.
Sensing what's inside.
But there are also closed-type EECs, like the enterochromaffin-like ECL cell found in the gastric epithelium. They don't have direct luminal contact, but still secrete important substances like histamine in response to other signals, like the hormone gastrin.
So some sense directly, others respond to internal cues.
Correct. And here's a truly fascinating historical nugget for you. Did you know the very first hormone ever identified, all the way back in 1902 by Bayliss and Starling,
No, what was it?
was actually a GI hormone, secretin, discovered because acidic fluid in the duodenum caused pancreatic secretion even when nerves were cut. Proof of a chemical messenger in the blood.
The birth of endocrinology started in the gut. Amazing.
It really is.
Our sources highlight several specific, crucial GI hormones, and understanding their individual roles is key to mastery, I think.
Absolutely. Knowing the players is essential.
For instance, gastrin. Where's that from and what does it do?
Gastrin is produced by G cells, which are EECs located in the gastric antrum, the lower part of the stomach. It's stimulated by the presence of oligopeptides, small protein fragments from digested protein, and also by parasympathetic nerve activity, like through the vagus nerve.
So protein in the stomach triggers it.
Primarily, gastrin acts via an endocrine pathway, traveling through the bloodstream to target ECL cells, stimulating them to release histamine.
The closed type cell you mentioned.
Exactly. And it also directly targets parietal cells in the stomach lining. The combined effect is stimulating the secretion of both histamine and hydrogen ions, protons, which means more stomach acid production.
Gets the stomach ready to digest protein.
Precisely. Then we have cholecystokinin, CCK.
CCK, I've heard of that one. Makes you feel full.
It definitely contributes to satiety, yes. It's released from eye cells, another type of EEC, mainly in the duodenum and jejunum. It's primarily stimulated by the presence of fatty acids and hydrolyzed protein, the breakdown products of fats and proteins.
So fats and proteins entering the small intestine trigger it.
Right. And CCK is a versatile messenger. It acts both through paracrine, local, and endocrine distant pathways.
Doing double duty.
It targets vagal afferent terminals nerves that send signals to the brain, which is part of how it influences satiety. It also strongly targets pancreatic acinar cells.
The cells that make digestive enzymes.
The very ones. So CCK stimulates pancreatic enzyme secretion, especially enzymes needed for fat and protein digestion. It also causes gallbladder contraction, releasing bile to help emulsify fats. And functionally, it acts to inhibit gastric emptying and hydrogen ion secretion from the stomach.
So it slows down the stomach while boosting digestion in the intestine. Makes sense.
Coordinates the whole process beautifully.
Next up is secretin, the first hormone discovered.
The historical one. Released from S cells, again, EECs in the duodenum, it's specifically triggered by the presence of protons, so by acidic chyme arriving from the stomach.
Acid triggers it.
Yes. Like CCK, it can act both paracrine and endocrine. Secretin targets vagal afferent terminals and, crucially, pancreatic duct cells.
Different pancreatic cells than CCK targeted.
Yes. Acinar cells make enzymes. Duct cells make bicarbonate fluid. Secretin strongly stimulates the pancreatic duct to secrete large volumes of water and bicarbonate.
To neutralize the stomach acid.
Exactly, protecting the duodenal lining and creating the right pH for pancreatic enzymes to work.
Okay, gastrin, CCK, secretin. What else?
Let's talk about glucoinsulinotropic peptide, GIP. Originally called gastric inhibitory peptide, but its main role is now known to be different.
GIP. Okay.
It originates from K cells, EDCs found mostly in the duodenum and jejunum. This hormone is stimulated by fatty acids and glucose in the intestinal lumen.
Sugars and fats trigger it.
Yes, it acts endocrinely, traveling to the beta cells of the pancreas.
The insulin-producing cells.
Right. And it directly stimulates insulin secretion. This is part of the incretin effect, the fact that oral glucose triggers a bigger insulin response than intravenous glucose because these gut hormones get involved.
Preparing the body for the incoming sugar load.
Precisely. A feed-forward mechanism.
Very clever. Any others we should highlight?
Two more important families often released together from the same cells. Peptide YY, PYY, and the proglucagon-derived peptides like GLP-1 and GLP-2,
PYY and GLP-12.
These are secreted from L cells, EECs found predominantly in the distal small intestine, ileum, and colon. Their release is stimulated later in digestion by the presence of fatty acids, glucose, and hydrolyzed protein reaching further down the gut.
So signals that digestion is well underway, or maybe that enough has been eaten.
Exactly. PYY acts through both endocrine and paracrine pathways on neurons and smooth muscle. Its effects are generally inhibitory downstream of where it's released, but also signals back up. It reduces gastric emptying, pancreatic secretion, gastric acid secretion, and intestinal motility.
Kind of like putting the brakes on, the ileal brake mechanism.
Yep, that's a perfect description. It also contributes significantly to the feeling of fullness, inhibiting food intake.
And the GLPs.
Proglucagon-derived peptides, particularly GLP-1, also from L cells, are hugely important. They act both endocrine and paracrine on neurons, pancreatic islet cells, and epithelial cells. GLP-1 is another major incretin hormone, like GIP.
Also boosting insulin.
Yes, stimulating insulin release in response to glucose, but interestingly, it also suppresses glucagon secretion from pancreatic alpha cells, and it slows gastric emptying and reduces appetite. GLP-2, derived from the same precursor, primarily promotes epithelial cell proliferation and gut growth, important for repair and adaptation.
So GLP-1 is key for blood sugar control and satiety, GLP-2 for gut health.
In broad terms, yes, and these are clinically very relevant, as we'll discuss.
Wow, a whole orchestra of hormones managing digestion.
It really is. Now, expanding on that, let's explore paracrine regulation, where the signaling is much more localized.
Right, acting on nearby cells, not via the blood.
Exactly. In paracrine regulation, a chemical messenger or regulatory peptide is released from a sensing cell, often an EEC or maybe even a nerve cell within the intestinal wall. But crucially, instead of traveling through the bloodstream, it acts on a nearby target cell by diffusing through the very narrow interstitial space between cells.
Like whispering to your neighbor instead of shouting across the room.
That's a great analogy. These paracrine agents can influence various cell types within the gut: wall smooth muscle cells, absorptive enterocytes, secretory cells, other neurons, immune cells.
Lots of local crosstalk.
Tons. A classic example is histamine, released from those ECL cells in the stomach lining.
Triggered by gastrin.
Right. Histamine then acts locally in a paracrine fashion on neighboring parietal cells, binding to H2 receptors and strongly stimulating stomach acid production. It works synergistically with gastrin and acetylcholine.
So multiple signals converge to boost acid.
Often the case. Another key paracrine example is serotonin, or 5-hydroxytryptamine, 5-HT.
Serotonin, isn't that a brain chemical?
It is, but actually the vast majority, something like 90% of your body's serotonin, is found in the gut.
No way. What's it doing there?
It's released from specialized EECs called enterochromaffin, EC cells, and also from enteric neurons and mucosal mast cells. It plays critical roles in regulating smooth muscle function, initiating peristaltic and secretory reflexes, and also influencing water and electrolyte absorption across the intestinal wall. It's a major player in gut motility and sensation.
So gut serotonin is crucial for gut function itself.
Absolutely. And there are other important paracrine mediators found in the gut wall too, things like prostaglandins, adenosine, and nitric oxide, NO, all capable of influencing GI function like blood flow, secretion, and muscle activity.
A lot of local chemical signaling happening.
Constantly. And it's worth noting again that some substances, like CCK, can act as both paracrine and endocrine regulators. It can stimulate nearby vagal nerve endings via paracrine action, action, sending signals to the brain, while also traveling through the blood endocrine to stimulate the pancreas and gallbladder. Shows the versatility.
That flexibility of some of these messengers is truly remarkable. Now let's dive into the neural control, the second brain.
The neurocrine regulation.
In its general form, neural regulation involves a neurotransmitter released from a nerve terminal in the GI tract, which then affects an innervated cell muscle, gland, blood vessel, or another neuron.
Signal transmission via nerves.
What's interesting is that sometimes there aren't direct one-to-one synapses, especially with smooth muscle. The nerve endings, called varicosities, release neurotransmitters more broadly into the vicinity of the target cells.
Like a sprinkler system rather than a direct hose.
Good analogy. This neuroregulation is crucial for both very localized control within a specific gut segment and for long-distance communication between different parts of the GI tract.
Coordinating activity across the whole organ.
Now, at the cellular level, the precision of how receptors recognize these signals, especially peptide hormones, is incredibly nuanced. You mentioned this selectivity is often influenced by post-translational modifications.
Right, changes made to the peptide after it's synthesized. This can dramatically alter its function.
Let's use PYY as a detailed example to really nail this down for mastery.
Okay, good example. PYY is released from those L cells mainly in response to fats and carbs reaching the distal gut. It's initially released as a 36 amino acid peptide. We call it PYY136.
The full-length version.
In this form, it can bind to multiple receptor subtypes, specifically the Y1, Y2, and Y5 receptors, which have different effects. However, circulating in the blood, PYY136 encounters an enzyme called dipeptidyl peptidase 5E, or DPP4.
DPP4. That sounds familiar. Didn't we mention inhibitors for diabetes?
Exactly. We'll connect those dots. DPP4 cleaves, or cuts off the first two amino acids from PYY136. This produces a shorter version, PYY336.
Just slightly shorter. Doesn't matter.
Hugely. PYY336 loses its affinity for the Y1 and Y5 receptors and becomes significantly more selective for the Y2 receptor. The Y2 receptor is particularly important in mediating the inhibitory effects on appetite and gut motility, the ileal brake effect.
Wow. So the presence of just one enzyme, DPP4, by modifying the peptide, completely alters its specific biological response, directing it mainly to the Y2 pathway.
That's an astonishing level of fine-tuning. A single enzymatic SNP changes the message.
And these intricate discoveries have real-world clinical implications. Let's revisit glucagon-like peptide 1, GLP-1.
Another key peptide hormone, also released from L cells in response to nutrients.
Also derived from the proglucagon gene, and also a substrate for that same enzyme, DPP4.
Correct. GLP-1 is absolutely crucial for glucose homeostasis. As we said, it powerfully stimulates insulin release from pancreatic beta cells, but only when glucose levels are elevated, it's glucose dependent.
So less risk of causing hypoglycemia.
Much less, which is a huge advantage. It also suppresses glucagon release from alpha cells, which prevents the liver from releasing more glucose. This dual action effectively lowers postprandial blood glucose.
So what does this all mean for you, the listener, in a clinical sense?
Well, this profound understanding led directly to groundbreaking therapies for type 2 diabetes, recognizing that GLP-1 is rapidly degraded by DPP4.
Lasts only minutes in the blood.
Researchers developed two main strategies. One was creating long-acting GLP-1 receptor agonist drugs like exenatide, liraglutide, semaglutide that mimic the effects of natural GLP-1 but are resistant to DPP-4 breakdown and last much longer.
Mimicking the natural hormone but making it stick around.
The other approach was developing inhibitors of the DPP-4 enzyme itself, drugs like sitagliptin, saxagliptin, linagliptin. These block the enzyme that breaks down your body's own natural GLP-1 and GIP.
Letting your natural incretins do their job for longer.
Exactly. Both approaches enhance incretin action and improve blood sugar control. These treatments have become widespread, not just for their effects on insulin and glucagon.
There's more.
Oh, yes. They also have significant extrapancreatic effects benefits outside the pancreas. They slow gastric emptying, which helps reduce the post-meal glucose spike and increases satiety. They act on the brain to reduce appetite and promote weight loss, especially the agonists. They improve overall insulin sensitivity in peripheral tissues. And some evidence suggests they may even help preserve the mass and function of the pancreatic beta cells over time.
Truly multifaceted benefits stemming from understanding gut hormone physiology.
It's a fantastic example of bench-to-bedside translation. Now, that covers endocrine and paracrine. Let's delve into the remarkably complex neural regulation of the GI tract.
Nerves. Extrinsic and intrinsic, you said.
Correct. The gut receives innervation from two distinct sets of nerves, the extrinsic nervous system and the intrinsic nervous system. The extrinsic nervous system refers to the nerves that innervate the gut but have their cell bodies located outside the gut wall.
Coming from the brain and spinal cord.
Essentially, yes. These are part of your broader autonomic nervous system, ANS, sympathetic and parasympathetic, and their primary role is to modulate the gut's intrinsic functions, kind of like higher command influencing local operations.
Okay, tell me about the parasympathetic side first.
Parasympathetic innervation mainly arrives via the vagus nerve, cranial nerve 10.
The wandering nerve.
Indeed, it's very extensive, and also via the pelvic nerves originating in the sacral spinal cord. The vagus nerve innervates the esophagus, stomach, gallbladder, pancreas, the entire small intestine, the cecum, and the proximal colon, a huge territory.
Covering most of the digestive tract.
Most of it. The pelvic nerves then manage the distal colon and the anorectal region. Now, consistent with how the parasympathetic system typically works, the preganglionic cell bodies are located in the brainstem, for the vagus, or the sacral spinal cord, for pelvic nerves. Their long axons travel to the gut,
All the way from the CNS.
where they synapse with postganglionic neurons. And crucially, these postganglionic neurons are the enteric neurons that reside within the gut wall itself, within the intrinsic plexuses.
Ah, so the extrinsic nerves don't directly talk to the muscle or glands.
Generally, no. The signal transmission nearly always occurs via a neuron within the intrinsic enteric nervous system. The synapse between the extrinsic preganglionic fiber and the intrinsic postganglionic neuron is what we call an obligatory nicotinic synapse, meaning it strictly relies on the neurotransmitter acetylcholine acting on nicotinic receptors. It's a very reliable relay point.
Okay, so parasympathetic signal comes in, triggers an ENS neuron, which then acts locally.
That's the pathway. Now, sympathetic innervation.
The fight-or-flight system's influence on the gut.
Right. Sympathetic innervation originates from cell bodies located in the thoracic and lumbar segments of the spinal cord. Their preganglionic fibers travel a shorter distance and synapse in prevertebral ganglia collections of nerve cells located outside the spinal cord but in front of the vertebrae, like the celiac, superior mesenteric, and inferior mesenteric ganglia.
So the synapse happens outside the gut wall for sympathetic.
Correct. Then the postganglionic sympathetic fibers travel from these ganglia to the gut. Unlike the parasympathetic side, these sympathetic nerves can directly innervate effector cells, particularly blood vessels, causing vasoconstriction to divert blood flow away from the gut during stress. They also innervate some glandular structures and can directly influence the enteric neurons within the plexuses, generally inhibiting their activity.
So sympathetic can bypass the ENS sometimes, especially for blood vessels.
It can. Now, both the parasympathetic and sympathetic divisions also carry afferent or sensory neurons back from the gut to your central nervous system.
Sending information back to headquarters.
Exactly. For vagal afferents, the cell bodies are located in the nodose ganglion near the brainstem. They project centrally to the nucleus of the tractus solitarius, NTS, in the brainstem, a key integration center. Their peripheral terminals extend widely into the gut wall layers. Spinal afferents, which generally run alongside the sympathetic pathways, have their cell bodies in the dorsal root ganglia alongside the spinal cord.
Sensing different things.
There's overlap, but vagal afferents seem more involved in monitoring physiological conditions, luminal contents like acidity, nutrient composition, osmolality, non-painful stretch. Spinal afferents are often associated more with noxious or painful stimuli, and also mechanical events. These peripheral terminals are found in all layers of the gut wall, constantly detecting a wide array of information.
A constant stream of data going back to the CNS.
A huge amount. And all the essential components of a reflex pathway, afferents, interneurons within the CNS or ganglia, and efferent neurons are present within this extrinsic innervation. These allow for long-loop reflexes.
Reflexes involving the brain or spinal cord.
Yes. A key example is the vagovagal reflex, where both the afferent sensory limb and the efferent motor limb are mediated by fibers within the vagus nerve.
All contained within the vagus.
Sensory info travels up the vagus to the NTS in the brainstem, synapses with interneurons, which then activate vagal efferent motor neurons traveling back down the vagus to influence gut function. These extrinsic reflexes are critically important in regulating GI function, especially coordinating responses to a meal.
Can you give an example?
A prime example is the gastric receptive relaxation reflex. When food enters the stomach and starts to distend it, stretch receptors are activated. Sensory signals travel up the vagus nerve to the brainstem.
Info arrives.
The brainstem processes this and sends signals back down the vagus nerve, causing the smooth muscle in the upper part of the stomach, fundus and body, to relax.
Why relax?
This relaxation allows the stomach volume to increase significantly to accommodate the meal without a large rise in internal pressure. It makes the stomach a compliant reservoir.
Clever way to allow filling comfortably.
Very. So, in general terms, while parasympathetic activation typically activates or stimulates GI processes like secretion and motility, with some exceptions like relaxing sphincters.
Rest and digest.
Right. Sympathetic activation tends to be inhibitory to overall GI motility and secretion, while constricting blood vessels and contracting sphincters. Sympathetic tone increases during stress or exercise, effectively putting digestion on hold.
Fight or flight takes priority.
Makes sense physiologically. Now that covers the extrinsic system modulating from the outside. Let's turn to the intrinsic neural innervation.
The enteric nervous system. The ENS. The second brain.
All those names fit. This is truly a marvel of biological engineering. It's unique because its nerve cell bodies, the interneurons, the motor neurons, the sensory neurons, the whole network are entirely contained within the wall of the gut itself.
A brain within the gut.
Pretty much. It consists of those two major plexuses we identified in the gut wall layers. The myenteric plexus, our box, located between the longitudinal and circular muscle layers.
Primarily controlling muscle activity.
Correct. And the submucosal plexus, Meissner's, which resides in the submucosa.
Focused more on secretion and blood flow.
And absorption, yes. These two plexuses are extensively interconnected by interganglionic strands, forming a continuous network running the length of the GI tract. The neurons within the ENS are functionally diverse, just like in the CNS. There are intrinsic primary afferent neurons, IPANs, that sense the local environment, interneurons that process information, and efferent motor neurons that control muscle contraction, secretion, blood flow, and even endocrine cell release.
So it has all the parts of a reflex arc locally.
All the essential components of a reflex pathway are contained entirely within the ENS itself. When stimuli in the gut, all chemical or mechanical, are detected by IPANs, they can activate local interneurons, which then stimulate or inhibit efferent neurons to alter function locally.
It can run its own show.
It absolutely can. The ENS can act quite autonomously, coordinating complex patterns like peristalsis or segmentation, largely independent of direct extrinsic input for moment-to-moment function. However, as we discussed, ENS neurons are extensively innervated by extrinsic nerves, sympathetic and parasympathetic.
So the extrinsic nerves set the overall tone or provide specific commands, but the ENS executes the details.
That's a great way to put it. The extrinsic nervous system modulates these intrinsic reflex pathways, adjusting the sensitivity or strength of local reflexes based on the body's overall state or needs. This incredible capability for autonomous function and complexity is precisely why the ENS is often called the second brain, or considered a key part of the brain-gut axis.
It really justifies the name.
It does, and in the clinical world, a congenital disorder called Hirschsprung's disease provides a stark and profound illustration of just how critical the ENS is for normal life.
What happens in Hirschsprung's?
This condition typically presents in newborns, often with a failure to pass meconium, the first stool, within the first day or two. This leads to symptoms of intestinal obstruction like severe chronic constipation, abdominal swelling, distension, and vomiting.
A blockage, essentially. Why?
The underlying pathology is the complete absence of the ENS nerve cells, both the myenteric and submucosal neurons in a segment of the distal bowel, usually the rectum and sometimes extending into the colon. These neurons fail to migrate properly during fetal development.
So that part of the gut has no intrinsic nerve control.
Correct. That segment of bowel cannot relax properly and cannot generate peristaltic movements. It remains constricted, creating a functional obstruction. Content backs up behind it.
That sounds serious. What causes it?
It's understood to be a complex polygenic disorder, often linked to mutations in multiple genes that are crucial for the normal migration, proliferation, differentiation, and survival of the neural crest cells that give rise to the ENS. The RET gene is a major one implicated.
It really highlights how essential that intrinsic nervous system is for basic gut function.
Absolutely. It's not just optional fine-tuning, it's fundamental.
What an incredible deep dive into the nervous systems of our gut. Extrinsic, intrinsic, vagovagal reflexes, the second brain. Okay, to really bring it all together now, let's think about the integrated response to a meal.
The symphony, as you called it.
Yeah, it's like a finely tuned symphony of GI function. Our body's response to a meal is classically divided into several distinct phases, right?
Yes, traditionally we talk about the cephalic, oral, esophageal, gastric, intestinal, often broken into duodenal, jejunal, ileal, and sometimes colonic phases.
The cephalic starts even before food enters the mouth.
Right. The mere sight, smell, or even thought of food triggers signals via the vagus nerve to start preparing the GI tract, increasing saliva production, gastric acid secretion, pancreatic secretion, anticipating the arrival of food, feed-forward regulation,
Then oral esophageal involves swallowing and transport.
propulsion down to the stomach.
Then the gastric phase kicks in when food arrives in the stomach, triggering local reflexes, gastrin release.
Acid secretion, mixing contractions.
Then as chyme enters the duodenum, the intestinal phase begins, triggering CCK, secretin GIP.
Pancreatic secretions, bile release, slowing gastric emptying.
And as digestion proceeds down the small intestine and into the colon, you get later signals like PYY and GLP-1.
The ileal brake, satiety signals. It's a continuous, overlapping cascade.
So if we truly connect this to the bigger picture for the listener aiming for mastery.
In each of these phases, the meal itself presents specific stimuli, chemical signals from nutrients, mechanical distension from volume, changes in osmolality. These stimuli then activate the different pathways we've meticulously discussed.
Neural pathways, both intrinsic ENS reflexes and extrinsic long-lip reflexes like vagovagal ones.
Paracrine pathways, local signaling via histamine, serotonin, etc.
And the humoral or endocrine pathways, hormones like gastrin, CCK, secretin, GIP, PYY, GLP-1 traveling through the blood.
And all of these pathways, activated by the meal, converge to produce precise and coordinated changes in the gut's effector functions.
Things like smooth muscle contraction for motility, secretion of digestive juices, acids, bicarbonate mucus, and regulation of absorption and blood flow.
Exactly. And crucially, there is considerable and constant crosstalk between all these regulatory mechanisms. Nerves influence hormone release. Hormones influence nerves. Paracrine signals modulate both. It's a highly integrated network.
Not just separate systems working in parallel.
Not at all. They are deeply intertwined. Just like maintaining homeostasis in other vital body systems like the cardiovascular or respiratory systems, controlling GI function requires these complex, dynamic regulatory mechanisms to continually sense the internal environment and orchestrate appropriate responses. It ensures everything works seamlessly for optimal digestion, nutrient absorption, waste removal, and overall health.
Wow, we've truly gone on an extraordinary journey into the incredible, intricate world of your GI tract today. From its distinct layers, mucosa, submucosa, muscularis, serosa,
to its astonishingly complex control systems, endocrine, paracrine, and the dual neural systems, extrinsic and intrinsic.
and its profound connections to other body systems, like the liver via the portal vein, the immune system, and the brain itself. We've seen just how much goes on behind the scenes.
Hopefully you feel you now possess a comprehensive understanding of this often underestimated organ system.
You now have a truly comprehensive understanding of the functional anatomy and the general regulatory principles of the GI system. This knowledge allows you to grasp the fundamental workings with the depth of a seasoned professional.
So, as a final thought for you to ponder, building on everything we've discussed, given the gut's remarkable autonomy, its intricate neural network, the second brain, and its constant communication with the central brain via nerves and hormones, how might our emotional state, our stress levels, our overall psychological well-being impact this second brain and GI function in ways we're only just beginning to fully appreciate? That brain-gut connection works both ways.
That's a fantastic question to leave us with, the interplay between mind and gut. Fascinating. Okay, that's all for this deep dive. We hope you feel incredibly well informed and ready to explore even more.
Keep digging deeper.
Until next time.